Hi, this is Miss Litton, and this is my Honors Bio wonderful class. Say hi.
Hi.
Okay, so first thing I want to go over is structure of the phospholipid bilayer. And let's make sure we have that down. Okay, so it's called the fluid. You've got to take your hood off, otherwise it makes you nervous. Fluid mosaic model. What's the fluid part of it? Wait, did I teach you fluid mosaic?
Yeah.
Is that AP level? It's very easy. The fluid part is, what did we say that a membrane is the consistency of?
Olive oil.
That's the fluid part of the fluid mosaic. Then what's scattered amongst the phospholipids? All of these proteins. That's the mosaic part. Okay? So you have your phospholipid bilayer. That is the layer that separates the inside of the cell from the outside of the cell, okay? And so you need to be aware of phospholipids. You need to know that it's amphipathic. That means there's a polar end and a nonpolar. The nonpolars are to the interior of the membrane. The fatty acid chains are to the interior. And then the phosphate groups are hydrophilic. They're either to the watery interior or the watery exterior, right? Because they're hydrophilic. Good. Okay, that's the phospholipids. Then the proteins, some are integral, where they go all the way through, or they're embedded in, and then some are peripheral, where they're just on one side, or on the outer edge. Okay? AP just took a test, and they're out there debriefing, freaking out.
Yeah.
Okay. And then we'll go over the protein functions. Now, in this picture, there is a lipid missing. What lipid is missing in this picture? Cholesterol. Yeah, so cholesterol and the, sorry, I didn't hit play. Cholesterol would be like 4 rings embedded in there, yes? And what does the cholesterol have to do with?
Keeps it from becoming liquid.
From freezing or falling apart. So there's one word to explain that, and that's fluidity. Okay, the fluidity of it. Okay, and then you have carbohydrate side chains. And those carbohydrate side chains are either glycoproteins or glycolipids. Okay, glycoproteins or glycolipids. So they're carbohydrate, but the carbohydrate attached to a lipid is called the glycolipid, and if it's attached to a protein, it's called the glycoprotein. So 3 of your 4 important organic molecules are found in the membrane itself. So phospholipid, there's one lipid. Cholesterol, also lipid, right? Protein and carbohydrates. Okay, you good on that? Yes.
So can you repeat that?
Sure. 3 of your 4 important organic molecules are found in a cell membrane. Phospholipids, and then another type of lipid is cholesterol, right, which is a steroid, but it's still underneath the umbrella of that. And then you have proteins, and then carbohydrates. And the carbohydrates are either a glycolipid or a glycoprotein, collectively called the glycocalyx. Okay, how do you feel about the structure of a cell membrane? Okay, so then the next thing we need to talk about is how you cross a cell membrane, right? So you can either go what?
Through the phospholipid. Not if you're large.
Okay, what is it? Sorry, never mind. Here we go. Through the phospholipid bilayer, but not if you're large or charged. You can use a channel or a carrier and whole membrane. Endocytosis and exocytosis. Perfect. Okay, so that's how you're going to cross. Now, the deal is you got to remember that the interior right here is hydro what? Phobic, because of those long fatty acid chains. So if you have something that's got a charge on it, it's going to not be able to get through that hydrophobic region. So then it's got to use a protein, or it's the whole membrane that has to work to get it across if it's got a charge on it. If it's too large, it can't fit through there because it's just too big. Now, what are some things that can go through the phospholipid bilayer? Easy peasy lemon squeezy. Water, for sure. But if you need a little assistance, what do you use? Aquaporin. Think water, aqua, right? Pour. It's going through the pore, aquaporin. Okay, so water can go through easy peasy. What else can go through easy peasy? Carbon dioxide. One more gas. Oxygen. Yeah. they can go right through the cell membrane. No problem. Water, oxygen, carbon dioxide. What do we call it when water moves back and forth across the cell membrane? Osmosis. Good. All right. Now let's talk about protein functions. And here you can see the cholesterol, right, that's in there. Okay, so the protein functions. The first one, it could be a channel. We learned this, yes? Channel. It could be a carrier. Now, when we talk about channels and carriers, okay, those could be used in facilitated diffusion, right? Where you have a molecule going from a higher concentration to a lower concentration, but it doesn't cost you any what? Energy. It doesn't cost you any energy. If it doesn't cost me energy, why can't it just go through the phospholipid bilayer?
It's either larger charge.
Larger charge. I'm getting a hot flash. Is it hot in here?
Yes.
Okay. Yes.
What's the main difference between a carrier and a channel?
Okay, so a channel is kind of like an opening that something can fit through, but a carrier, it physically binds to it at some point. Like literally carries it. You know, a channel, I can just open a door for you and say, come on through. A carrier, I'm carrying you across the threshold. Does that make sense? Okay. All right. And then if channels are not used for active transport because they're just open, but carriers can be used for active transport, right? And that's if you go against the concentration gradient. If you're going from a lower concentration to a higher concentration, if you're shoving even more things to one side of a membrane, then it's active transport. What's the most popular, yes?
Oh, I know.
What is the most popular pump? That's when you use a carrier. Yes, sodium potassium pump. And this is where it's binding and it's concentrating sodium to one side and potassium to another side. Okay, we good on that? All right. Then, okay, so we said channel, carrier. Then we said cell recognition. So these are going to be primarily glycoproteins. And this is how you tell one cell from another cell, both within the tissues of your own body, but also with something that's not from your body. If you have some sort of pathogen coming in, it can recognize that. Even if it's a tissue that you surgically had put in, like, oh, I know I need a heart for a heart transplant or I need a kidney, if it doesn't have the ID of your body, that's like your cellular fingerprint, if it doesn't have the idea of your body, then your immune system is going to start to do what? Attack it. Yeah, exactly. Okay, channels, carriers, cell recognition. What's the next one? Receptors. So receptor, it could be a receptor for a hormone or some other signal to turn that cell on, turn that cell off, okay, to open up another channel, sorry. So receptors, like all proteins, are very, very, very specific. Okay, and then your next one is, so we said receptor, then what? Enzymatic. So it could catalyze a reaction that is needed inside the cell, but it could be usually connected to a receptor, which then triggers an enzyme that's right next to it. Okay, like adenylate cyclase. And then your last one was junctions. Okay, so your junctions, they could be very tight. They could be two channels hooked up together. They could just be two proteins cemented together. But those proteins are holding our two cells together. Now, that becomes really critical in animal cells because the animal cells don't have a cell wall. All cells have cell membranes, right? If it's alive, it's got a cell membrane. It just may or may not have a wall. Okay, ask your bio buddy, how are you so far on all of that? Yeah. Okay, so let me back up. Okay. So, guys, there's a level here that I haven't gone into yet with you with G proteins and adenylate cyclase, but it is an enzyme. Enzymes make reactions go faster. So this is something that facilitates a reaction, And if you don't have that enzyme working, if that protein is not working well as an enzyme, then the reaction's not going to go forward. Okay? All right, so then we already went over this. 3 ways to cross the membrane. Okay? And then we talked about both passive. Do you need me to hit this one again, or are you good with that? You can identify all 3 if I asked you? Okay. All right, and then permeability. Sorry about that Y right there. Okay, so I already did that. Okay, I'm tired. Today feels like a Friday to me. Is anybody else feeling the Friday?
Yeah.
I so feel the
It feels like a Monday.
I'm so
I
delirious
feel
I
like a
can't even
Monday.
keep track of the days of the week, so you're doing better than me.
But just think, Friday is a half day.
Oh, yeah. Wait, is this the only
So
one?
technically right now, we're halfway through it Tuesday. Technically, since we get out a half day early on Friday.
I'm sorry.
See? Halfway through Tuesday. Okay. All right,
We also get
yeah.
off early on Monday.
Go.
Can you explain the junction proteins again?
Yes, I'm not holding you accountable for all the different junctions that there are, but you could line up 2 channels. So if we were 2 different cells and your channel lined up with my channels, things could pass right from one cell to another. Or just straight up just proteins that are in there on the edge, the 2 proteins could line up and stitch them together. Like your bladder, you don't want your bladder to leak, right, into your abdominal cavity. So it has tight junctions, real tight between the cells so no fluid gets out. And then there are ones where part of the cytoskeleton is shared, but I'm not holding you accountable for all the different varieties there. Okay, anything else on that? All right, now this we already hit as well, passive versus active transport. Is this the only kind of active transport over here on the far right? No. Okay, what's the active transport that's not shown in this diagram? Well, way to play. Way to play. You can't win if you don't play, so good. All right.
I should tell you.
Whole membrane is missing. Whole membrane. Right? That's when you have the whole membrane. Yeah, if your whole membrane is engulfing or spitting something out, that's active because you're moving cytoskeleton around in order to make that happen. All right, so passive transport requires no energy. You're going from a higher concentration to a lower concentration. If you're not large or charged, you can just go right through the phospholipid bilayer. Like we said, water, CO2, O2, they can go right through the phospholipid bilayer, no problem. Okay? If you are large or charged, you cannot go through the phospholipid bilayer between the phospholipids. You have to use a channel or a carrier, but it is still passive because you're going from a higher concentration to a lower concentration. Good. Active transport requires the movement of a protein some way to concentrate a substance on one side or the other, like concentrating sodium to the outside and potassium to the inside. Okay, and that's a sodium-potassium pump. That's one where I said 30% of your ATP is dedicated to that, right? Okay, so that is one example of active transport, but so is endocytosis and exocytosis, another example of active transport. Okay, ask your bio buddy, how are you feeling on this? Are there any additional objectives in this chapter? There's not. Okay, and then can we move on?
Yeah.
So let's talk about going through the phospholipid bilayer with water, and what is that called? Osmosis. So remember, osmosis has 3 components. The diffusion, so that's going high to low concentration. Diffusion of a solvent, which is usually water. So it's diffusion of water across a semi-permeable membrane. So if I just have a bucket and I put sugar in it and add water, is that osmosis? No. Why isn't it osmosis? There's no membrane. You got to have a membrane involved. If you're going to talk about osmosis, you have a membrane. Now, typically it is a cell membrane. But when you want to model a cell membrane, that's when you use dialysis tubing or something like that. You're modeling a cell membrane. In this case, we're just modeling a cell membrane. In your lab, you didn't model. You used a real cell membrane. What cell membrane did you use? The egg, right? So we got rid of the shell, and then they soaked in vinegar, right, to get rid of the shell, and then they were in corn syrup for 3 or 4 days. And the corn syrup was what? Hypertonic or hypotonic? Hypertonic, and water must flow from the hypo. So water left the interior of the egg and went out into the corn syrup. Yeah?
Yes.
Okay, so much so that there was like water at the top of the corn syrup, so when you went and got the egg, it's like the sugar was all on the bottom, right? Where did that water come from? That water came out of the egg, yes? Okay, so then I gave you these eggs that were all shrivelly. And where did you put those shrivelly eggs? In water. And it was not tap water even, it was distilled water. So it was as hypotonic as you could be. And water must flow from the hypo, so it would go into your what? Egg. And so the mass of your egg would increase every 15 minutes when you measured it, yes? Okay. Now, in this situation, if we label this side A and this side B, okay, remember our phrasing. If something is hyper means you have a lot of it, okay? So which side has more solutes? B. So this side is hypertonic, and side A would be what? Hypotonic. But remember, it's all relative, right? Remember in class, I said, what if this membrane what if there was another membrane here, some more water, and there was a lot of sugar in it? And if this one was C, then C would be hypertonic and B would be hypotonic. But B is still hypertonic relative to A, right? It's all relative. It's like, look at the membrane, look on either side, right? When you have something like this, you can't say left and right when there's 3 of them. Right? You have to look at that, okay, A to B. A is on the left, B is on the right. But if you look at B and C, B is on the left and C is on the right. Do you see what I'm saying? So it's always relative one to the other. Yes?
Would the hypertonic solution want to go into the hypo to where it's even? Or
Would it want to? Absolutely.
But to it can't.
Absolutely. No, in this case it couldn't because it was a disaccharide. But in your lab, let's talk about what happened in your lab, okay? In your lab, you had a beaker. What did I put in that beaker? Iodine. I put iodine in that beaker. What lab? I don't know what you're talking about, Melissa. Okay. So here's the iodine, yes? And then inside there, I put a bag, and what went inside the bag? Yeah, I had glucose in here. I'm just going to put glue for glucose, okay? And starch. Do you agree with that? And originally the bag started out white, yes? Okay, and then what happened to the bag over time?
It started to fill with the iodine and turned blue.
It started to turn blue, okay? Now, why did it turn blue? Yes, so iodine, remember you asked me, would the sugar want to move? Somebody asked me, would the sugar want to move? Yeah, the sugar wanted to move. The iodine wanted to move, right? It went from a higher concentration to a lower concentration. How do we know it did that? Because we know when iodine and starch hook up, the starch turns what color? Blue, dark blue. Okay? So iodine definitely went in. Did starch want to come out?
Yeah.
Yes, it did, but starch is too big. If it could get out, how would we have known that it got out? Yes, all out here would have started turning blue, right? But it didn't. It didn't turn blue, so therefore starch did not leave that bag. Yes?
Then on the other end, when the egg is in water, the sugar leaves. So then is there still
No. The sugar didn't leave. Now let's stick. We've got 2 different labs. I'll go back to the egg in a minute. Can we come back? Put a pin in that. We'll come back to the egg in a minute. I want to finish talking about this particular lab. Are we okay about the iodine and the starch?
Yes.
Okay. Who's our third player?
Glucose.
Where was the glucose originally put?
In the bag.
In the bag. Did the glucose come out? Yes. How do you know the glucose came out? Yeah, you use those test strips. Remember, they started off like kind of a pale blue, and then when we tested them in directly into glucose, they went kind of a greenish or a brown. So this bag, glucose could move out. Glucose is a monosaccharide. When I was showing you this right here, this isn't glucose. What is this? Sucrose. Sucrose is a disaccharide. Glucose plus fructose. So it was just a bit too big. So it's probably wanted to go out, but couldn't. Okay? Couldn't. Okay, now back to this, and then I'll go back to the egg. Come back to the egg. Yes. Yes.
How do we know whether glucose or iodine is bigger?
Oh, okay, so glucose is C6. I gave you the formula. Glucose is C6H12O6. So you have 6, 12, and 6. What does that add up to be? 24. Iodine is IKI. It's 3. 3 atoms.
So glucose is larger.
Yeah. Okay, what else? Yes.
Would you also argue that one is larger than the other because of the amount of time it took to...
No, I wouldn't do it on time because charge could affect it and other things as well. You can talk about whether, you know for sure the biggest molecule in this experiment was what?
The sugar.
Not the sugar. The starch. How do you know starch was the biggest?
Because it couldn't fit.
It's the only thing that couldn't move. Yeah, it's the only thing that couldn't move was the starch. Yes?
How was the iodine able to get through the bag?
Because it's so tiny. The holes. So glucose can get through. Glucose is C6H12O6. What does that add up? How many things? 24, right? Iodine is IKI. You don't need to memorize that. It's just 3 atoms. So it's smaller. It's a lot smaller than glucose. Yes?
Did the glucose leave the bag?
Okay, back to that. Did the glucose leave the bag?
Yes.
Yes. How do you know the glucose left the bag? Test strips.
Like, is there no glucose left?
Oh, no. Do you think it went all the way out of here? No. Did all the iodine go into the bag? No. Why wouldn't that happen? Because it wants to be the same. Yeah. Okay, anything more on this? Okay, then let's go back to the egg. Okay, so in the situation there, that corn syrup, okay, water left the egg and went out into the corn syrup because water is very, very small and it can move. The corn syrup didn't move into the egg. Yeah, the egg just got smaller. Okay, what else? Okay, all right, so let's keep going. So we understand that both of these have solutions in it, but one has more solute in it than the other, right? So if we again said A, and B, side B is hypertonic to side A. And it doesn't, so I just don't want you to think that if for something to be hypotonic, it can't have anything in it. If something's hypotonic, it's just less than whatever's on the other side of the membrane. Okay? Then we recognized what would happen to cells if we put them in different solutions. So, for instance, if I had 3 beakers right here. Whoa. Okay. All right, no problem. I don't know what's happening, but okay. So let's say I cut myself. Cutting. Okay? Don't do that. Don't do that. Don't do that. It was an accident. Whoops. Okay. So I have some red blood cells here. Okay? So these are my red blood cells. Okay? Predict what would happen. Okay. Sure. Okay. Water is splashing. There you go, water. Be there. What would happen to each of these red blood cells if I said this solution was hyper, I said this one is iso, and I said this one was hypo. Okay, in your mind, just take a minute, think it, don't say it out loud, just in your mind, what would happen to those red blood cells if I put them in each of those solutions? Now whisper it to the person next to you, not too loud. Okay, If the solution around it was hyper, do I need to put stuff in here or no?
No.
It's a hypertonic solution. Solution. Not cell, but solution. What does hypertonic mean? There's a lot. So this one would have a lot of stuff in it. If this one is isotonic, does that mean nothing in it? No, it doesn't mean nothing in it because this red blood cell has stuff in it, right? So I need something to be about the same, yes? And if this one is hypotonic, do I need to put anything into it? No. Okay. So what would happen to this red blood cell right here if it was in a hyper? It would shrivel because water must flow from the hypo. Sorry. Okay. Now there's 3. Okay. So those red blood cells will shrivel. If it's in isotonic, it would stay the same. And if it was in a hypotonic, what would happen? Yeah, it would burst. Okay. Because water must flow from the hypo. Okay, so then we saw that. Here's isotonic, red blood cell, plant cell. Here's hypertonic, water must flow, it's shriveling down. And then hypotonic, it's swollen up. Okay, then take a look right here. I didn't want to draw it this time because that worked out poorly. So I actually went and got a cucumber, okay? And so if I have this garden, I have all these cucumbers, what kind of solution do I need to put it in if I want to have pickles? Hypertonic, because water must flow from the hypo, and then I could get a jar of pickles. Does that make sense? Okay. All right. Then next, using a protein. When I use a protein, it could be both active and passive. Passive, I'm going with the gradient. Active, I'm going against. Okay, good job. And then we talked about endocytosis and exocytosis. Endocytosis means coming in. Exocytosis means going out. And there are 3 types of in. Don't say them out loud. See if you can just think them in your head. The 3 types of endocytosis. Think them in your head. Okay, what is cell eating called? Phagocytosis. Good, is cell eating. What is cell drinking called? Penocytosis is cell drinking. Then the last one, it's got a couple names to it. Do you remember what the last one was? Receptor-mediated endocytosis. In receptor-mediated endocytosis, you have a bunch of receptors. Molecules bind to those receptors, and then it does endocytosis. Okay, so it knows it has everything it needs. Okay, and then that was it. Do you have any questions you would like to ask me? Yes, sir.
So, I don't know for sure if you confirmed it, but I checked the calendar and it says page 64.
Last chapter had no how to suggest this one.
Oh, this one does. You know what? I wrote the quiz already. Can I just leave it off?
Yeah.
Or do you want me to rewrite it?
No.
I will rewrite it if you want me. I'll get you to the book. Hashtag good choice. Okay. Are we good with that? Because I already wrote it. Okay. Any other questions? Yes.
What is the receptor-mediated?
Receptor-mediated, see, these are proteins that are receptors. So imagine them not there originally. Okay, so imagine they weren't there. They were just receptors. Like, where did that one come from? So they were just receptors. Then they came, what are they doing? Okay, and then they bound, and then the cell went like this. It would not trigger the endocytosis, the cytoskeleton moving, until all the receptors were bound to a compound. Does that make sense? Okay. What else? It's what she's talking about right now. Yes.
Is there a set amount of the receptor proteins on each thing? Or are they like all the same?
This is probably, what you're seeing right here is not a good representation. There would probably be a whole bunch more receptors in that patch or area before it did that. Yeah. Okay. What else? What else? Are you good? Do you want to do a TikTok?
Yes.
All right. Have a wonderful evening. Don't stay up too late. I love you.